Okay, so thank you everyone for joining this webinar. So as Marisa said, I will elaborate on how we in De Laval develop and evaluate cleaning solutions for milk installations to help achieve good milk quality. So who exactly is De Laval? Well, De Laval is a market leader that provides integrated milking solutions for farmers. We are part of the Tetra Laval group, which consists of 3 industry groups, being Tetra Pak, Sidel, and De Laval. And together with these 2 companies, we cover the full value chain from cow to consumer. I would like to show you a short video now to summarize who we are and why we believe we can help in making a difference in the dairy sector. So let's hope it works.
We take it for granted, but milk makes superfoods. It's your day's intake of a whole range of vitamins and minerals. It's protein, it's energy. So you might think a kilo of cheese isn't all that important, but it is. The world's population is growing and we don't know how to feed everyone. If we really want to be serious about this, we need to look at high nutrition foods. Milk and milk products are among those foods. And at De Laval, we need to constantly work to make sure it's produced more efficiently. And this isn't just a matter of us doing business, it's a matter of sustainability. How can we do more with less? Every time we help a farmer find a way to produce more milk from the same herd, we're making food production more sustainable. By introducing a new working method on the farm, a new parlour, better liners, better hygiene, better data to make better decisions. By cutting disease and keeping cows healthier, by increasing their milking lifetime. Every time we do that, we help farmers do more with less. Every time we do that, we're moving on the road to more sustainable food production. And we'll never stop. We can't stop. Innovation is in our blood. It's been in our blood since Gustave de Laval patented the centrifugal cream separator back in 1878. That's almost 140 years of continuous knowledge accumulating year after year, changing how we do things, innovating to make them better. We're the market leader and a trusted partner for thousands of farmers around the world. There's 4,500 of us serving over 100 countries. Add to that 1,000 dealers selling our products and advising farmers across the world to make the best decisions for their farms. And then there's the farmers. Millions of farmers around the world. A lot of livelihoods. A lot of people depending on milk to live. We're farmers too, you know. We know a thing or two about the challenges and the charms on the ground, in the field. It gives us an edge. It helps us lead. It helps us innovate. In short, we live milk.
Okay, so let's go to the next slide. If it works, yeah. So at the MQAH department of De Laval, our primary emphasis is on ensuring high quality milk and optimal animal health. And this is achieved through practices such as maintaining udder hygiene, hoof hygiene, and thorough cleaning of milk installations. Within MQAH, we have a research and development team that is based in Kansas City in the US, where new safe and effective cleaning detergents, hoof, and other hygiene products are being developed. Technical service is part of that team, and it consists of 5 people, including myself, that are located in both North America, so in the USA, and in Europe. At technical service, we perform clinical trials of potential MQAH solutions. So essentially, we serve as the bridge between research and development and the practical implementation of our products in the field. So we test all products on the farm, we assess their potential, and we ensure that they meet all necessary criteria before they are introduced to the market. So we often conduct cleaning trials to evaluate cleaning detergents, which I will focus on further on in this webinar. Besides that, we also have a wide range of teeth disinfectants, and before the launch of such a new teeth spray, we perform teeth conditioning trials to test its effect on the teeth skin and the teeth end. We also assess its spray performance, and we monitor the milk for potential residues such as iodine. And then we also have some other clinical trials, such as hoof care trials and innovation trials. In addition to clinical trials, we also evaluate new technological tools that are available on the market. We provide training on milk quality and animal health, and we actively participate in scientific publishing. So before we go deeper into the cleaning trials and the cleaning evaluation, it's important for you to understand the basics of cleaning of milk installations. So the initial question is then, why exactly do we clean? Well, as a consumer, your primary expectation is to have high quality milk, and not only because it's the end product, but also because it is the basis, or it can be the basis for various other milk products. You want your milk products to smell, look, and taste good, but you also want them to be free of debris, chemicals, and contaminants. However, people tend to forget, or sometimes tend to forget, that this is milk, and milk comes from cows that usually live on farms. Our ideal scenario would be a farm looking like this, but the reality is that in a less well-managed farm, it can easily resemble this or even this, which will then lead to cows with dirty udders. And it's obvious that this will impact the milk, and thus also the quality of the milk. There are various points in the milking process that will determine the final milk quality, and one of the important contact points is the milking machine. So inadequate or improper cleaning of the milking system can then result in situations like these, and that is exactly what we are trying to prevent. So cleaning includes both the removal of any unwanted material left after milking and the killing of microorganisms. In order to clean, we need to understand what exactly we are cleaning, since the different steps of the cleaning process target different types of deposits. So milk is composed of many ingredients, and its composition can vary based on factors like the breed, the feed, and the lactation stage. Water is the main ingredient, and the remaining components are carbohydrates, so mostly lactose, fats, proteins, and minerals. Milk fat is insoluble in water, but it melts at temperatures above 40 degrees Celsius. So that means that if we clean at temperatures lower than that, it could result in a greasy film on the outside of, or on the contact surfaces. Milk proteins, such as casein, they can form deposits that have a rainbow shine to them at low levels, and when they further accumulate, they will look like a varnish brown staining. And then the minerals that are present in the milk, but mostly those that are present in the water that is used for the cleaning, can also deposit on the milk lines. And they can compromise the efficacy of the cleaning detergents. So therefore, water quality and water hardness are important factors in farm management. So milk is sterile at secretion in the udder, but it gets contaminated by bacteria even before it leaves the udder. So usually these bacteria are at that point harmless, such as lactic acid species, and sometimes they can even be beneficial, like lactobacillus. But then during milking, handling, and storage, milk can be further infected with bacteria that can reduce the shelf life of the milk, or that can cause milk spoilage, such as Pseudomonas or coliforms like E. coli. And this last one is also a pathogenic species that, just like other ones, such as Listeria and Campylobacter. And then besides bacteria, there are also some fungi like Aspergillus species and yeasts like certain Candida species that can be present in the milk. Apart from the soils of milk, there is also milk stone, and this is actually the result of different layers of minerals, fats, and proteins and fats that are accumulating on the pipeline or in the tank, and that will then result in this hard and whitish buildup that is more difficult to remove. And then lastly, we have biofilms. So these are structured aggregations of microorganisms that are attached to the surface and are embedded in a protective matrix called the extracellular polymeric matrix, or EPS. The exact composition of this matrix that differs greatly between different microorganisms, and also between the growth conditions under which the biofilms were formed. but generally they include hexapolysaccharides, nucleic acids, and proteins. So biofilms are very complex and highly organized structures that have different microenvironments in them, and they can form entire communities, almost like miniature cities. They thrive in flowing and wet systems where there are plenty of nutrients available. And that means that milking systems, and especially automatic milking systems that have a constant milk flow at 37 degrees Celsius, provide a very good environment for them to grow. So there is a wide range of bacterial species, including Pseudomonas and Listeria, that can form biofilms, and also non-biofilm forming bacteria, fungi, and viruses can be encapsulated in the biofilms. We also know that there are some bacterial species, such as Microbacterium, that have been proven to be the key species for the formation of biofilm. So what happens in biofilm formation is that on these imperfect surfaces or on deposits, especially in low flow areas, bacteria can easily attach to these surfaces. And over time, they will start excreting the EPS, which will then firmly attach them to the surface. And they will start growing and form microcolonies. And once that biofilm becomes too large, or during events such as cleaning, some pieces of the biofilm might break off, which will then result in bacterial contamination. And these pieces can also form new biofilm growth elsewhere in the system. The problem of one of the most difficult things about biofilm is that they are more resistant to cleaners and disinfectants, and that is partly due to their protective matrix, so due to the EPS. It makes it challenging for chemicals to penetrate the biofilm, and a lot of the active ingredient is used up simply by reacting with the microbes and the outer layers of the biofilm. And thus it's very difficult for the cleaners to really get into the core of the biofilm and disintegrate it completely. So it's easier to prevent biofilm than it is to cure them. This brings us to how we clean. So cleaning of milk installations is done via cleaning in place, or CIP. And this is a method of cleaning the surfaces of pipes, process equipment, and milk bulk tanks without disassembly, which means that the equipment is cleaned by pumping or spraying water onto or through product contact surfaces. The approach for cleaning depends on the type of milking system that is used on the farm. So we typically make a distinction between conventional milking systems and automatic milking systems. In conventional milking systems, such as, for example, like you can see here, the parallel parlor and the rotaries, their cows are typically milked 2 to 3 times a day by the farmer on fixed times, and cleaning is then started once all cows are milked. In automatic or voluntary milking systems, the cows go to the robot when they need milking, and they are milked one by one. And like I said before, this means that the milk will be flowing through these lines continuously, and we typically clean there every 8 hours. Despite these differences between these milking systems, the actual cleaning process remains quite similar, but I will come there in an instant. So there are 6 key principles of cleaning. These are interdependent, and they play a vital role in the cleaning process. So all of these 6 parameters need to be there in order to have a good cleaning. So first, there are the detergents. These are the acids, the alkalines, and the sanitizer that help in the removal of the soils and in the reduction of microorganisms. Then we have the water, where both water quality and water quantity are important. We also need to use adequate temperatures, depending on the type of soil that we are going to remove. And then during the cleaning, we also need mechanical energy. And it's also important to keep in mind the contact time of the cleaning agents with the milking system. And then finally, we also need personnel to set correct cleaning parameters and to perform regular maintenance. In Europe, we typically use a 3-step cleaning process. So it begins with a pre-rinse to remove residual milk with lukewarm water, so that it is above the melting point of the fats, but below the burning point of the proteins. Next, there is a wash cycle. This is done at high temperatures to dissolve fats and proteins during approximately 8 minutes. If we want to remove some inorganic minerals, then we typically use acids, and this is done in alternate cleanings. And then the cleaning process ends with a final rinse with cold water to remove all chemicals with potable water. In other parts of the world, a 4- or a 5-step cleaning process is more common, where they incorporate an additional wash with a sanitizing product. So in our cleaning trials, we observe the performance of the cleaning detergent over a span of several months on the farm. So this involves conducting weekly or bi-weekly cleaning evaluations to closely monitor its effectiveness. So during the cleaning process, we monitor all cleaning parameters to make sure that the system is operating optimally. After the cleaning, we then conduct a thorough evaluation of the cleaning efficacy of the detergent, and therefore we use various methods. Sorry. So our evaluation begins with a visual inspection, where we examine the crucial parts of the milking system to identify any potential fouling, corrosion, or wear on system components. This is then usually followed by ATP swabbing, which is a method where we measure ATP, so the energy molecule that you can find in all living cells. And we do this also on different parts of the milking system using a luminometer. And this technique serves as an indicator of organic matter that is still left on the surfaces. However, this ATP can also react in the presence of cleaning agents, and thus sometimes it's not that straightforward to interpret. Depending on the nature of the cleaning detergents that we are testing, we might also take some samples for detection of possible biofilm growth. And this is done by plating them out for performing a total bacteria count. And then finally, in the last year, we've also tried to incorporate the Bactiscope into our assessment technology. And I will elaborate further on this one in the next slides. So the Bactiscope is a device that uses high-intensity wave alternating UV light that reacts with the outer protein shell of most bacteria, which is called the S layer or the surface layer. So there are 4 separate UV lights of different frequencies, and when they merge, they will bounce off the double protein shell, and this will then result in a typical green turquoise glow. And that allows us to quickly detect the presence of bacteria and biofilms. The colors under the Bactiscope typically range from dark blue, which is usually dust or dirt, to turquoise and green, which are then the contaminants. And I was just talking about the Bactiscope, but actually EIT has 2 products that can do this. So we have the Bactiscan, which is a large flashlight basically with a wide, powerful beam that enables us to see large areas at once. And then we have the Bactiscope that you can see here. And this is a camera equipped with the special UV light on a probe, and the probe is available in different lengths. And this can be used to inspect pipes and hard-to-reach areas. And basically, for our milk installations, that is the one that could be the most useful. And so the last year, we have been collaborating with EIT to test and optimize the Bactiscope for our industry. In the upcoming slides, I will present some examples of what we typically observe compared to how it appears under the Bactiscan or the Bactiscope lighting. So on farms, there are flies, especially in summer. And if the milking system is close to the barn, then it's not unusual for all the equipment to be covered with fly stains. So when you examine this under the Bactiscan, you can see these stains light up in a distinctive turquoise and blue color. These two pictures display the lid of a milk bucket that is used for milking specific cows whose milk need to be separated from the rest of the herd, for example, when they are on antibiotics. So it's evident that this lid hasn't been cleaned for some time because we can see a lot of milk residue and we could even see some fungi. And when we examine it under the Bactiscan, then we see that there were both these green and red lighting. So we're not completely sure what exactly the red color is, but we think that they might be fungi. Here you see some pictures of part of the milk bulk tank on a farm where there was poor milk quality. So the farmer had, or actually the milk, had very high coli and total bacteria counts. So the two first pictures are of the inlet and the outlet of the milk bulk tank. And you can clearly see some vivid green spots, especially at the edge where it is sealed with the rubbers. And on the right side, there's a picture of a valve rubber, which is open or closed based on the milking system status. And this rubber also displays some soiling at the edges, but it's mostly blue soiling. And then on top, there are some little dots that are green, which actually reveal some sign of wear and tear of the rubber. These two pictures show the inside of the claw ball of a milking cluster. So actually this piece, and that is where the teat cups are connected to the cow during milking. So this part of the milking system is constructed from a whitish transparent plastic, and under the Bactiscan or Bactiscope, it illuminates with a white-blue color, but this makes it more difficult to detect soiling on this type of material. So this slide also illustrates the difference between the Bactiscan and the Bactiscope. The Bactiscan emits stronger light, which makes it particularly useful for observing larger areas at once, whereas the Bactiscope is a probe and is more suitable for detecting biofilms inside pipes. So here you can see a video of what the Bactiscope can pick up inside pipes. So you can see some green marks at the top of the pipe. You can also see that the liquid will light up, but it is especially the case when the liquid is soiled or when it contains detergents. So the Bactiscope has both a special UV light, but it also has a normal white light that can be adjusted to optimize the visualization of soiling. This example shows the inside of the milk receiver, which is where all the milk is collected before it's being sent to the milk bulk tank. So at first we couldn't see anything remarkable until we used the Bactiscope, and there we can clearly see a blue mark under the inlet of the receiver, which is probably a result of mineral buildup of the hard water that was used on the farm. So it didn't result in any high ATP values, which makes us think that it's not any buildup of biofilm. And we saw on the same farm a few months later, we saw this, the same actually, but a little bit more advanced. So you could see that there was a little bit of brown staining. But then if you use the Bactiscope, you could really see it very clearly around the circumference of the entire receiver. And this is at the water level. So, and this brings me to the last slide of this presentation with a short summary of our main findings of the Bactiscope. So the Bactiscope is a fast screening method for the detection of possible biofilm and residues in pipes. We found it to be best to use it in combination with ATP swaps or total bacteria counts to further specify the type of soiling that we are visualizing. We also found that we could access a lot of points in conventional milking systems. However, for automatic milking systems, the head of the Bactiscope is still too big, as these pipes are generally narrower than those in conventional milking systems. And finally, we observed that there is a delicate balance between the UV light and the white light of the Bactiscope. So it's crucial to fine-tune this balance depending on the pipe materials and the width of these pipes. For instance, stainless steel pipes will reflect more light. So there it could be better to reduce the white light a little bit compared to in plastic pipes. And this emphasizes the importance of adjusting this Bactiscope settings accordingly. And that actually brings me to the end.